Good morning, everybody. My name is Ryan Murphy. I'm in the University of Minnesota Urban Forestry Outreach and Research Lab, and we are here for our October virtual MnSTAC forum. This is a forum series that is put on in collaboration between the University of Minnesota and the Minnesota Shade Tree Advisory Committee. This month, we have Kent Honl to present on treatment timings for disease and insects. Kent Honl is an arborologist with the Rainbow Tree Company and does a lot of outreach and education in addition to that through venues such as online webinars, speaking at the Minnesota Shade Tree Short Course, and also as an instructor teaching arboriculture at Hennepin Technical College. So with that, Kent, I will yield the virtual floor to you, and thank you for being here.
Oh yeah, well, thank you, Ryan, and greetings, welcome to everyone that I know and that I don't know out there. Thanks for your time today for me to talk about treatment timings for disease and insects. If everyone, I don't know how many people have been up in the fire tower at Itasca, but that's where my picture is from for this opening slide here. Just kind of big, broad overview kind of intro slide is what I wanted to get across there. But as Ryan said in the intro, I'm Kent. I've been in arboriculture since 1994, working at Rainbow Tree Care based out of Minnetonka, Minnesota. And that arborologist title is just kind of made up within Rainbow. There's a handful of us that are focused on technical training and research projects. I view it as kind of like spanning across the academic research to reach practitioners, you know, kind of being the bridge across the two. Because if research doesn't get applied in the real world, well, what use is it, you know? And oftentimes those are siloed enterprises, the people out in the field doing stuff, you know, they're not talking directly to researchers, and researchers are doing their thing. So, you know, it's the right and the left hand of our industry and our trade. So I'm in that privileged position to be able to kind of translate the research and put it into practice, merging science and practical application. I've got my board certified master arborist qualification and the track certification. Enjoy doing all this stuff. You know, I get to talk to people about trees all day for a living, and it's great. And as Ryan pointed out, I serve as an adjunct faculty at Hennepin Tech College, teach arboriculture course there. I've got 10 students right now. That's a lot of fun. And I've been doing that since 2014 up in Brooklyn Park on that campus. So today, the whole subject is treatment timings. And really it's based on, informed by Integrated Pest Management, which we just generally call IPM as a shortened acronym for it. And what that is basically is just preventing pests from reaching economically or aesthetically damaging levels with the least risk to the environment. So it's not about complete eradication of pests, it's managing them to fit within thresholds that are tolerable and constantly working to reduce the off-target damage of that. And how we do that with IPM is things that I'll go into, like monitoring, forecasting methods, and the goal is accurate timing of treatment, because what you end up with then is a reduced use of chemical products. If you don't spray every 3 weeks all summer when you don't need to, like, it saves you money for one thing, and it's a lot less chemical load in the environment, and you have more effectiveness of the treatments. You know, if you're hitting the pest or disease at just the optimal time, that's what we should be doing. So when you start off, say you're looking at a list of pests or diseases, like at Rainbow, we've got a few hundred different insects and fungi that we treat. You know, it's just this overwhelming array of different bugs and things to track. So, you know, it's really necessary to develop some kind of a systematic approach to tracking your treatments. This is a snip out of the treatment guide. You know, it's kind of an outdated one because our database that we had, we're using a different one now, Arbor Gold. So that column kind of just to the right of the middle doesn't exist anymore. But this is, my treatment guide, like European elm scale and what some of the treatments are for it and the timing, et cetera. And then, I've got degree days and all that stuff. But basically, this is just a spreadsheet that helps keep track of what the different protocols and the treatments are. So without some kind of a systematic approach, you're just going to get lost, really. And when I was in the midst of, 10 or so years ago, starting off, when this was handed over to me, okay, now Kent, it's your job to track when all the treatments are going to go. And I really had that deer in the headlights look, but I kind of stepped back and realized I based a lot of my approach to arboriculture off from this German forestry professor from 200 years ago, the guy Friedrich Pfeil. And as it happens, the word Pfeil is the word for arrow in German. And it's kind of like the arrow from this guy, Friedrich Pfeil, is pointing me in a certain direction of how to manage trees and care for them. And he said, ask the trees how they want to be cared for. They'll teach you better than books. He was very experiential, hands-on, empirically based forestry professor. And so my teaching and my practice, I try to really base it a lot off from his direction. So where that led me to was like the trees themselves providing a pattern for how to organize the information about them. And I realized like, okay, the tree itself kind of becomes a template that I can populate with the knowledge of the different insects, and I thought, okay, I'll just map out all the insects and diseases that I'm managing onto a branching pattern. And it really made sense for me where I could go, okay, one branch is fungi, the other branch is insects, and then there's another branch that's the abiotic or non-living conditions. And if you go further down the branch about the insects, we can branch them into sucking insects, borers, chewing insects, and that's not like Coleoptera, Lepidoptera, like scientific classification. It's more like from the practical approach of what do they do to the tree, because the way you treat them kind of falls into a similar pattern according to what they do to the tree. And then if we're going down that branch of the fungal conditions, it breaks down into foliar diseases, decay fungi, cankers, vascular diseases. So what we'll end up looking at mostly today is the protocol patterns for the sucking insects, borers, chewing insects, foliar diseases, because decay fungi, we're not really treating those. Cankers, you're not really treating those. Vascular diseases, yeah, we do preventive treatments on those, but that's not our main focus today. The main thing I'm pointing to here is like from the beginning, having some kind of a systematic way to map out everything that you're dealing with so it becomes less overwhelming and you can sort of fit it into some reasonable way to approach what you're up to. So here's a whiteboard that I drew out one afternoon. I just put pretty much everything on this branching key and fit it in there. And it gave me a far less overwhelmed feeling about what I was up to. Like I've got it all the way out to, okay, there's the caterpillars, that's canker worms, and there's white mark tussock moth, out in those. And if I go out into the fungi, there's the leaf infections, there's the anthracnoses, etc. So it just makes sense. It's an example of ask the tree how it wants to be cared for. It'll tell you better than any books. Just using this branching pattern to organize your information. I mean, that and or spreadsheets, it all kind of fits together. And keeping in mind, like what our goal is to aid in the tree defense. And part of IPM, really important part is that we use pesticides, but use them responsibly as part of this toolbox, that it's not all about pesticides, but when you need to use them, you use them responsibly, and they can fall into spray applications or systemics that we'll talk about, or there's a whole array of root flare injections that could be pesticide things, or it's a lot of fungicide based things too for root flare injections or macro infusions. Because what we're up to on behalf of the tree is stepping in to aid the tree defenses to protect against energy depletion. If you look at this graph, this is out of Shigo's work. You can see where that arrow is pointing. There's a steep drop in reserve energy that falls in the spring, right when the reserve carbohydrates from last year are getting mobilized to build that new crown of leaves. And then once you've got a crown of leaves, the photosynthetic area on the tree starts rebuilding that energy budget within the tree. But that sharp drop, you'll notice, that's when your IPM, your plant healthcare activities just start going crazy, because all the stuff happens all at once. That vulnerable leaf tissue is easy for chewing insects to attack. It's easy for spores of fungi to land on there and infect it, etc. So what we're doing with our responsible pesticide use is protecting the trees during that little vulnerable period in there. And looking at this too, I can look at that little dip. It kind of looks like a pulse or a wave on a chart. I'm really a believer in the pattern language of the world, like that there's a pattern in the branching of trees, in tributaries of watersheds. Branching is one pattern. Another pattern in the world is waves, pulses, repeating occurrence of phenomena that you can read in the world. So there's a whole other talk, Ryan, have me come back sometime about reading the pattern language in the world. So I'll park that. I won't go off on that tangent too far. Main thing here is when that energy drops, we're stepping in to aid in the tree defense and protect against energy depletion. Now, bear with me with the toggling on these slides, it gets a little goofy. Okay, so another big concept in integrated pest management is this disease triangle. And I almost always find a way to fit disease triangle into any discussion of pest management because it's so pivotal. It sounds so basic as to be useless, but when you examine this, there's power in the simplicity of it. Really what it's pointing to is that you have to have the interaction of these three factors for a disease or an insect situation to arise, like an emergent property. It doesn't have an independent existence in the world until a host within a given environment that's conducive to a pathogen getting activated brings about the condition for a disease to exist. So that's why I call it an emergent property. It emerges out of the interaction of these factors. So that opens up a lot of avenues for us to intervene. I was just talking about the responsible pesticide use. That's where we'd be down controlling the pathogen. But we shouldn't ignore, there's other places within that triangle to be active in controlling pests or managing them too within thresholds. Like, can you change or modify the host? Is there some way to increase the vigor or the strength of the host? Or maybe you're going so far as to remove that host and replant something that's resistant or immune to the pest that you're dealing with. Or within the realm of cultural practices, can you change or modify the environment, like using mulch or proper irrigation, a whole host of those things. So you can change any or all of those elements within that triangle. And we don't have to think about it as the disease triangle. You can put the emphasis on the health triangle. If you're managing the environment and the host, there's probably less you're going to have to do with controlling pathogens. So look at it holistically in that regard. So if we just start with a concrete example here, apple scab, understanding the life cycle of it, how that works. That you have an infected tree, those scabs on the leaf and the fruit is going to drop to the ground, that sits there, and the next spring, as the moisture shows up, then these spores start drifting up and infect the next year's new tissue, etc. It goes back and forth like that, spreading around in the tree. So if we're following this example, applying the disease triangle model to it, in midwinter, for example, like say it's January, that host, we know it harbors the scab fungus. It's a crabapple. Maybe even if you were there on the site in the middle of the summer, you saw the yellow leaves with the brown spots on them, premature leaf drop. But the environment in the middle of the winter, it's cold, dormant, probably snow on the ground, nothing is active, because the pathogen at that point is just overwintering in leaf litter. So at that point in time, there is no disease yet. It's we've accounted for what's going on in the triangle, but it's not the conditions set to make the disease show up. But when spring arrives, then all the elements are in place for an infection to occur because the environment corner has changed. Temperature is starting to go up. Moisture starts to increase with spring rain. The host becomes receptive. And here's the important part for monitoring for IPM. The leaf emergence, it brings out vulnerable tissue. When it just is coming out, like between the size of a mouse ear to a dime or so, that's tender tissue where when that pathogen, the spores down in that leaf litter start to germinate and drift around, that can drift up and land onto that vulnerable soft tissue. It hasn't built a waxy cuticle over itself yet to resist the invasion of a fungal spore on there. So now you can have that emergent property of apple scab fungus disease showing up in place because of the elements all coming together into the right configuration, basically. Which is why monitoring is so important for our timing of treatments in IPM. We got to find out what's the rate of a leaf development, like on the bur oak here, those new leaves coming out. If you're dealing with oak anthracnose, you'll want to know, have the leaves started to pop yet? What size are they? Or what's the level of a pest population? If we're checking for spider mites here, monitoring is just, sets of thresholds that you've kind of mapped out that you're checking for. You run a route of places around your region where you're working to make sure you know what's going on with the sets of trees that you manage. And monitoring is going to be combined with forecasting. If we're continuing to go with this example of apple scab, Cornell University has really mapped out quite in detail what it takes for getting apple scab infections to get going, the wetting period required for that scab infection. And you can see the temperatures in Fahrenheit go from top to bottom, and then left to right, it goes, how much time a leaf has to be wet at that temperature to produce a light infection, moderate, or a heavy infection. So you can see down on the bottom row, 33 to 36 degrees Fahrenheit, you still could get an apple scab infection, but you'd have to have 48 hours of continuous leaf wetness for the fungus to be favored enough to produce that. A heavy infection would happen if you had 96 hours of continuous leaf wetness at 33 to 36 degrees. So usually that doesn't happen. But when I see that bottom row, it points to like, it goes a lot slower when it's colder. That's why you put, your turkey sandwich in the fridge. It slows down the rate of biological activity. But there's still things that could happen. Like you leave your sandwich in the fridge for a week or two, you probably shouldn't eat it. It slows it down, but it doesn't stop it unless you freeze it. But in this case of the scab fungus, notice how as the temperature starts to go up, the amount of time it takes with a wet leaf decreases steadily until you get right into that sweet spot of 63 to 75 degrees. So a light infection, all it takes is 9 hours of leaf wetness if you're in that temperature range, 63 to 75. And if you get 18 hours, that could be a heavy infection. Now, further interesting to note, it's not completely linear. You get up beyond that sweet spot, get to 78, and the time starts to expand again, 13 hours at 78. But usually what we're dealing with in the spring, if you look at your weather forecast and you know that your leaves are opening up on the crab apples, they're getting into that roughly dime-sized range, and it's going to rain, it's going to be wet for 12 hours, and it's going to be in that temperature range, then it's time to start sending out your fungicide sprays to deal with that, to protect that tissue. Again, what we're doing is stepping in to protect vulnerable tissue where the tree doesn't have its own defense to do so. So with foliar fungicide applications, it's a whole chunk, now on your spreadsheets or your branching pattern, whatever you've set up, you still have to figure out, okay, what's the protocol? How do I know when to spray it, how many times to spray it, all that. Basically, like you can take a whole chunk out of that spreadsheet and say, well, anthracnose, tar spot, apple scab. They're all foliar fungi applications. They're all kind of going to follow the same protocol. The aim is to protect that new leaf surface when the conditions favor the infection. And the general pattern is going to be put the first spray on there at 1/2 to 3/4 leaf expansion when there's a forecasted event that could produce an infection. And then you repeat at 14 to 21 day intervals for a total of 2 to 3 applications, depending on how long the weather continues to favor those conditions for infection. And fungicide residual activity tends to be, you'll be good if you're hitting it every 14 to 21 days. So that's kind of the template for a foliar fungus application. And, you might have 15 different things that you now just sort of have templated out. They just go at different times based on when the host development is happening. Then with conifers, it's the same thing, but instead of leaves coming out, you're getting needles, so this picture shows the varying levels of leaf or needle emergence, I guess you'd call it. It's foliage. The one on the left there, that would be a sprayable spruce at that point. Like if you're talking Larula needle blight or Rhizosphere needle cast, that middle one still maybe has a little bit of bud cap left on it. Over on the right, those buds haven't even popped open. So there'd be no point in spraying that. Really all we're after is protecting that new tissue that's coming out on the spruce on the left. And what clients need to know, the spray has to be done at the right time. Like if they call you up and say, okay, I'm ready to spray my crabapple now. I just got back from Arizona and saw your notice in the mail. Well, we're not going to come and do that right now. It's too late. It'd be a waste of our time, the chemical, your money, etc. But you can get them on the list for next year. And clients a lot of times will ask, well, I've sprayed this thing year after year after year. Haven't we cured it yet? When can I stop spraying? Well, the treatment for things like scab has to be repeated annually for control, because really a new crown of leaves, it's starting from zero again. Not like apple scab will kill the tree, but most people are doing it for more of an aesthetic purpose. But it's not like this produces a cure. It's just annually stepping in to break that infection cycle, because the new leaves are vulnerable every year. The other thing too, with integrated pest management, we've moved away from spray equipment that shoots really high up in trees. Like for most of these foliar things, we can treat the lower 30, 35 feet of a tree. And that's sufficient for most cases with foliar fungi, because it's mostly on the shaded lower interior of a tree. And the upper sunny, wind ventilated area of a crown of a tree doesn't tend to get much like anthracnose or scab anyway. So that's part of how IPM is reducing its footprint, is like limiting yourself to just spraying the lower interior part of a tree. So now if we shift our focus over to the insect pests, there's a couple different categories of pesticides we'll look at, like sprays, and then there's systemics, which have quite a few advantages. With some historical context, going back to the early 1990s, if we had two-line chestnut borer in an oak, for example, what that required was spraying Dursban on it 3 times per season in the summer. And Dursban is something that you can't even have anymore. It's no longer labeled for use. But in the early 2000s, systemic insecticides came on the scene and enabled us to greatly reduce our pesticide use. And I know that systemics get a lot of bad press, like because of exposure for pollinators, and we'll talk about that. But when you consider what used to be the case of spraying trees and the inevitable drift that happened, using a systemic hugely reduced the overall footprint of what pesticides were going into the environment. This is actually a picture. Like, I started at Rainbow, I was a plant healthcare tech, and I hated myself going out shooting chemical up into the air into oak trees, 60, 70 feet. It was just terrible. Because there's no way you can spray a tree that big without getting off target drift. And those large trees are just really challenging to treat if you have to do with sprays and the challenges with scheduling. You know, everything has a very specific management timing, as I've been talking about timing. You know, if you're spraying your apple scabs and everything else, everything happens at the same time. Right when that energy dip happens with the trees, that's when all the pests attack. They've evolved to hit the tree at its weakest point in the cycle. So that's where you end up with these management challenges, you know, that you have to go back and do multiple site visits, because a lot of the products, spray products, have a limited residual activity. So they don't last that long. You have to keep going back to hit them. And if it's too windy or rainy, you can't do sprays at all. So the more we can move away from spraying trees, the better off we are in terms of reducing our footprint, and our operational headaches get greatly reduced if we can just go to systemics. Now, we don't yet have a systemically applied fungicide we can put on the soil and treat against apple scab. I mean, there are other things I could go into about systemic acquired response, but that's a whole other subject too. So I'll just park that so I don't go too far on that tangent. But, you know, to bring it back to the systemics and the advantages, you don't have off-target damage through drift. There's nothing going in the air, it's just going in the soil. And you can have a real accurate application. Like with a spray, you're just spraying a tree till it drips, and that could vary widely depending on, if it's a little bit breezy, you might have to apply more and get more drift in order to get the target covered. With a soil applied, you know exactly how many milliliters of stuff has to go into the ground. And the great thing, best thing probably about systemics, a lot more flexibility of timing. You don't have to be there right on the day when it's at the most vulnerable. You can put them in ahead of time. On the rainy day, the windy day, it doesn't matter. And you got a whole range of uses for shorter and longer term activity, and a wide range of pests can be controlled. So the focus is on pest here, not on pathogens, because we're not there yet with fungicides that work through soil injections quite yet. So what we do have in the toolkit falls into these three categories: acephate, dinotefuran, or imidacloprid as soil applications. Each one of them has its own rate of absorption, duration of effectiveness, and the list of insects it control. Like, you use the acephate if you want a really quick uptake time, like 3 to 7 days, that gets into the tree, and then it lasts for 30 to 40 days and it's gone. You know, that can be very advantageous if you want something that's going to get a quick uptake and effectiveness, but then be out of the pollen stream for next year's flowers. It's no product there to get in the way and cause trouble for pollinators. So a good IPM practice is to prescribe these effectively for what you need to have in place. Dinotefuran, longer uptake time, 2 to 3 weeks, but you get a longer duration of effectiveness. And then imidacloprid, 45 to 60 days, but then you get a full year of control of what you're working to deal with in terms of the insects. So there's, of course, I'd mentioned this pollinator concern. You know, it's of huge importance nowadays. So things like lindens, crabapples, magnolias, black locust, we just will not treat with the longer term systemics, because you'll end up having the product getting into the pollen and the nectar of those species, and flowering plants within the treatment zone. People like to put hostas, daylilies, that kind of thing around the base of their trees. Then we can't do those, because that will also have an uptake that will be injurious or lethal to bees. So the labels actually state that nowadays too, that they require application timing when bees are not foraging on plants, or just not even using that particular product if it has a long enough residual to affect the flowers for the next season. Now I get on to growing degree days. How's my timing? Yeah, good. 11:30 here in DC. I know there's interest in this and kind of seems like mystery surrounding it. Like, what are they? How do you use them? But I've been using these for about 10 years now, and really I'm a believer in them. And what they basically are is just a forecasting methodology where you can use temperature to guide your predictive ability of when a particular insect is going to be likely to attack. So I put the focus here on insects because this has nothing to do with fungi at all. With the fungi like scab that we looked at, what you're looking at is the emergence of the vulnerable tissue, what's the temperature going to be, and what's the moisture forecast going to be. So that's a whole its own constellation of factors. But with insects, since insect development is so tightly linked to temperature in the environment, degree days can really give you an almost mechanical ability to predict when they're going to show up. And though the term is called growing degree days, it's not really a measurement of a day. Think of it more as a unit of warmth that's occurred, that's accumulated during a season. And each insect has a really consistent threshold. Like when you reach a certain number of accumulated units, then say the sawfly is going to show up, or Japanese beetle or Magnolia scale, those kind of things. And how a degree day unit is calculated is you take the high and the low temperature for a given day, add them, and divide by 2. You're just averaging the high and low. And then it's subtraction of a base temperature, which is typically 50 degrees Fahrenheit. Other times people will use like 32 as their base, but generally speaking, 50 Fahrenheit is the base temperature used for growing degree days. And this year I've started to do a tracking in Celsius as well, because I really like the metric system, and it's the language of science. So why has the US so stubbornly refused to get on metric and everything else with the rest of the world? So it's kind of silly that we're still in Fahrenheit, but that's where we are. So I've got my chart set up in Fahrenheit and Celsius, but basically that's your growing degree day calculation. Average the high and the low for a given day, subtract 50, and that gives you a number. And that number is added to every number for that season that preceded it. So it gives you a cumulative total of those units. And here's a snip of my tracking chart. And everyone wants to know, like, well, where can I go online, or what kind of an app can I use to track these? And you can find loads of degree day information online. And sure, there's probably apps for it, but I'm kind of like, Fred Flintstone basically, and I want to have more of my own direct relationship to it. And the other thing where I've run into frustration in dealing with online figures for growing degree days is a lot of it is built for agricultural purposes. So it's all full of stuff about corn and soybeans and this and that, and ads for, different kind of herbicides, just junk that I don't need. Really, I just want a number for my locality. And the other thing, a degree day website is just going to give you what your degree day figure is for today's date. And then it's not going to allow you to forecast out a week, 10 days, 2 weeks, whatever. So that's why the past handful of seasons, I've just built my own spreadsheet with a calculation in it where I can input the forecasted figures into this thing and it does the calculation. So I can put forecasted temperatures out like 10 days and then see where we're going to potentially be, and then go, okay, I think the Japanese beetles are going to be out about 2 or 3 weeks, but with your degree day figure that you just pull off from some website, it just tells you what it is for today. And you're not going to be able to tell, like, well, with the forecasted weather, where is that going to land us? So this is basically built, you can see on September 15th, the high was 77 and the low was 52. Add those together, divide by 2, subtract 50, you get 14.5 degree days for that day. And then when that's added on to everything that happened previous in the season, that gives us 2,923 growing degree days. Now, an interesting thing too, as we're moving along, you can see September 24th, we're at over 3,000. And as of today, right now in late October, we're bumping up against 3,400 growing degree days. And we haven't seen that since 2012. I can remember 2012, it was 78 degrees on the first day of the shade tree short course. So normally we top out at around 2,700 growing degree days. So we're getting into way longer seasons. You know, any climate deniers out there, I can point to my own charts and show like, yeah, spring starts earlier and fall arrives later. And, you know, I've got my own tracking charts that I can point to for that. But, you know, this is basically pointing to like, I don't really do apps or websites for this. I put it into my own chart, you know, for my own relationship to it and my own ability to track and forecast. So bringing it into the actual categories, you know, for my branching pattern that we looked at earlier, I mapped it into chewing insects, sucking insects, and borers. So what does that look like? You know, with the chewing insects, they're this whole group. They have mouth parts adapted to consume leaf tissue, you know, that fits them because it's easier to attack than other parts of the tree, like twigs and bark. That's doesn't have a lot of nutrients. Leaves, that's high in chlorophyll, nitrogen, all kinds of good stuff, even some sugars, you know, that the leaf is making. So that's a jackpot if you can attack that part of the tree. And one of the main chewing insects that we've got locally is the Conifer Sawflies that can lead to rapid irreparable damage, you know, because once you strip out the needles off from a conifer, it's not going to grow back needles in that spot. So you end up with that kind of poodle tail look on a pine tree where sawflies have had a big infestation and ripped through there. So when your degree day chart is approaching 240 degree days, you know, it's almost mechanical, like 240 degree days shows up and my phone rings and it's arborist from Plymouth saying, oh, yep, we got it on Mugo Pines here. And then it's Maple Grove. And then it's, you know, just boom, 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 boom. And it's like, yep, we were hitting 240 and I knew it was going to hit right about then. Now, granted too, this is, keep in mind, it could be a week ahead of 240 and you'll get the first hatches in some areas because there's microclimates. And from the south to the north of the metro, it will vary. Like Lakeville will start getting earlier reports than you will in the northern suburbs. And 240 just means like you should be ready to, you know, have a peak infestation showing up by then. But be ready for the early reports of little 1/8 inch sawflies. Like by 240, they might already have been growing, feeding, so they're a 1/2, 3/4 inch long and causing some damage. But by 240, like it's happening. So for a treatment protocol, you know, the way that we sort of lump the foliar diseases into a similar approach, that's kind of like you can throw that same net over chewing insects. Like you use permethrin, general insecticide to just knock down the current population to keep them from spreading too much. And then you follow up with a longer term systemic like imidacloprid. So you put that in the fall, then next year, your next year's sawflies that come out are going to be controlled by that. I mean, provided you're not dealing with some kind of flowering tree, you know, that would affect the bees. So Japanese beetle, that's the other major chewing insect that we get. Just through observation, I've seen like the lindens finish blooming at 980 degree days on the Fahrenheit scale. And lindens are like the big magnet for Japanese beetle, but also for bees. Bees go crazy over those flowers. They smell great. And it's just a lovely time of the year to walk under linden trees and smell that. And the Japanese beetle adults show up at around 1100 degree days. That's a pretty tight timing. So we don't want to be spraying permethrin, this general insect killer stuff on linden trees, because sometimes those adults start showing up while the lindens are still blooming a little bit. You know, it's just a pretty tight window there. So, you know, we have to shift to a different insecticide. I put the note here, dramatic but not deadly, because I'm going to make this into a contrast to another insect later. What I mean by dramatic but not deadly is that Japanese beetle are highly visible. Their feeding creates visible damage that's just alarming. So that gets the phone ringing. It's a dramatic onset and result, but it doesn't cause the death of the tree, but it sure does get the phone ringing for people. So when we come to borers in the talk, I'll talk about deadly but not dramatic as the contrast here. But I wanted to point out acelepryn is a relatively newer insecticide with a lower environmental impact. So extremely low toxicity to off-target animals like birds, fish, and bees, and very effective on Japanese beetle when you apply it as a foliar spray, and you get a long residual, so you don't have to keep hitting it over and over. Like one spray will take care of you for 28 days. On lindens and anything else like that, it's great. You know, what it does is paralyzes the chewing muscles of the Japanese beetle so they can't feed. So it's just not even something that bees are going to pick up on or get affected by, really. So, you know, it's products like this are really a breakthrough for what we're up to using IPM friendly things. So on to sucking insect. Magnolia scale is one of our main ones. It's a challenging pest to treat successfully. Typically, you have to have a combination of treatments over multiple seasons. And IGR spray, what that means is an insect growth regulator spray. Again, it's where we used to use permethrin, just this broad spectrum insecticide to knock down the crawlers. Now we're using this insect growth regulator. What it does is it stops their development at the crawler stage, so they can't mature and become a new scale that sets up shop on another twig and starts to suck out plant juice. They're just arrested in their development, and then they can't grow and they just end up dying. So it really leaves other organisms unaffected, because, you know, other things that affect it, like ladybugs that are going to come and feed on crawlers, they're already adults. So, you know, that's not going to interrupt any process they need to go through. Or bees, you know, they're not even going to be coming around there because there's no flowers at that time. You know, basically the IGR sprays have really been developed for the IPM toolkit, and they're very successful. I've seen it work very well on Magnolia scale crawlers. Systemic soil applications, you know, your acephate can be good, but I don't want to, you know, we don't want to use imidacloprid on magnolias anymore because of the flower, you know, lasts a whole year in there. We don't want to have that. I see there's stuff going into the chat, but maybe we can cover that when I get done, because we're coming in towards the finish line fairly soon here. Now check out this slide. I actually have this as a video too, but I don't want to show it as a video because you'll just have the creeps all day long when you see the crawlers moving around in the video. But if you bring a magnifying lens, you know, like a loop or something like that, that you can look, because these are like the size of ground pepper on a twig of a magnolia. That's tough to see with the naked eye, but when your degree day chart starts hitting 2000, actually start monitoring in the 1900 range, but you'll really start seeing them at 2000 extending into 2800. That's a pretty wide window, and it's referred to as a prolonged asynchronous hatch. Prolonged just means a long time. Asynchronous means they hatch not at the same time. You know, they take the time and spread it out. This usually lands like from mid-August into September is what that falls into in the Twin Cities area. So I was talking about insect growth regulator sprays. That's what we put down for Magnolia scale crawlers, and it worked really well. So other monitoring, you know, there's conifer spider mites. There's kind of like two shoulders. They're a cool season mite. So we're looking at spring from 95 to 360 degree days, or fall from 2375 to 2800. So in the Twin Cities, we're already past that. You might still find conifer spider mites. If you go out and do this paper shake test, you just put a white sheet of paper under a spruce branch and shake it and see if you get 10 or more mites on a sheet of paper. And that's kind of, if it's above that, probably want to enact some treatment program. If it's below that, let the ladybugs and lacewings, other kind of creatures come and step in and, do their biological control on them. Now the borers, here's where we get to deadly but not dramatic. Stuff like the bronze birch borer, two-line chestnut borer, emerald ash borer, they're flying around, laying eggs that hatches, the larvae tunnel under the bark. You don't really see it happening immediately, but it has a deadly effect on the tree. So it's not something that gets the phone ringing in the way that Japanese beetle does. And the client might say, well, my tree looks fine right now. I'll wait and see what happens. Well, people aren't attuned to necessarily doing the preventive treatments before things get bad. People usually tend to want to treat the alarming things that are not deadly. But this is where a conversation comes in. Really, this is something you need to be dealing with ahead of time before it gets bad. So actually this bronze birch borer on the birch here, if you set that right next to an EAB, they look pretty much the same. They're just a different color. Now I remember this was probably 5-6 years ago. I was saying to a group of arborists, you probably never see the adult bronze birch borer in your whole career because they're flying around up in the canopy laying eggs. And then 2 hours later, this insect lands right in front of my face on a birch tree. So I called up Jeff Hahn at U of M Extension and texted him the picture. And he said, yeah, that's got to be a bronze birch borer. And I said, well, it just made a fool of me because I said, you probably never see that. But there it was right in front of me. Nature's always looking for a way to make a fool of you. So the borers, they feed under the bark. They're difficult to detect. They're shielded from enemies and pesticides, direct application, you're not going to be able to spray them. Like I said earlier, that's what we had to do in the 90s before we had systemics, but we've got better ways to go at them now. Whether it's the soil applications, root flare injections, our mainstay for EAB prevention is root flare injections with emamectin benzoate. There are trunk sprays for a handful of them like Zimmerman pine moth, but we don't do a ton of that. And the timing for most of these for treatment of borers is whenever the tree is actively growing, you need a canopy of leaves on it for the operation of a root flare injected material or soil that's not frozen for the systemics. So that makes the timing much less exacting than if you're doing like a foliar spray for fungus on the leaves, that kind of thing. So now I get to wrap it up here, bring it into the home stretch, right about on time. I get to talk about proactive versus reactive treatment. That most of what we've been talking about is reactive. There's already a pest that we're dealing with. And by that time a pest has attacked, you're just playing catch up to a situation. And a reactive treatment is going to produce a short-term result against a specific pest or a disease. Like you're just treating for apple scab when you spray that fungicide. You're just, treating for Japanese beetle when you spray a saliprin. That's why I put Rocky on here. He's already been punched in the face. It's too late for him to bob and weave or slip that right cross that just caved in his face. So what if we're putting our focus more on proactive treatments? That proactive treatments, you don't have to wait for the pest to have attacked already or established a history of a pest problem. You can do it anytime ahead of the pest attack because action's faster than reaction. You can do that before problems have occurred is always better. And the great thing too is you're going to get a non-specific result. You're strengthening the host against a whole array of pests. And that's going to be a longer term benefit to the trees you're dealing with. So things like the soil remediation with the air spade, you can incorporate organic matter and biochar. If you've looked at any of the work of Glyn Percival in England, he points to biochar and willow mulch and other things like soil amendments as producing acquired response in trees that increase vigor and resistance against pest attack. He's seen reduction in foliar fungus 40 to 60% in some cases with no use of fungicides at all. So, this really is the frontier of arboriculture, is if we can reduce our pesticide use as much as possible, and build up the resistance of the tree without the use of a lot of chemical things. That's really where we need to be going. Okay, so to review and wrap it up, IPM, there's a whole bunch of tools that increase our effectiveness and reduce off-target damage to the environment. Some of that is monitoring and forecasting methods with degree days and thresholds, knowing when is the time to treat, or when it's, okay for beneficial predators to come in and deal with it. And the protocols and the whole array of things you're dealing with can be less overwhelming if you classify them into the groups of pests and how that builds treatment protocols, and you can map out your information in a way that makes sense. And for me, that's always been the mapping it on a tree kind of template. It just makes sense that way. And then, I always like to put in a plug for the distinction between reactive versus proactive treatments. And I'm really just trying to move more towards an emphasis on proactive treatments. So yeah, now we've still got 9-10 minutes left. Can dive into the questions. Stop my share. We'll scroll back through if I can catch up. Wow, we've got a lot of people out there. Oh, does the application height apply to rhizosphere as well? Yeah, generally, if you can get that lower part of the tree, you usually tell, like the upper part of a spruce tree doesn't tend to get the foliar fungus. Same like for your Austrian pines with Sphaeropsis, Staphystroma, all that kind of stuff. So it can be difficult if you've got retired clients who are sitting at home looking out the window and they see you only spraying the lower part of the tree, and then they say, well, I paid to have the tree sprayed, but hopefully the conversation has happened ahead of time, that you're going to tell them, like, yeah, we don't need to spray that upper part of the tree necessarily. But yeah, with the conifers, it's the same kind of a situation. Okay, given unintended effects of spraying fungicides, how is the research going to address the reasons the trees are vulnerable to the fungi? Yeah, exactly. That's why I put that whole thing in at the end about proactive treatments. Like there's a whole realm of research and discovery that can happen. Like, what if we're favoring life below ground in the tree with fungal networks that then support epiphytic fungi and bacteria within the tree that are antagonistic to the pathogenic fungi that live in the foliage? That's, I think, where Glenn Percival's work comes in, that there's all sorts of pathways that where, increasing organic matter, the capacity for biochar to host different bacteria and other fungi, probiotic, basically, like that's where, we won't have to use fungicides as much, or if at all, if we're doing all the other things like managing the host and the environment properly. So yeah, I'm glad that question was put up in there. Okay, Lauren, how often does the information you're coming up with relate to the high, low, safe, risk periods, major tree diseases? Oh, yeah. Yeah, with oak wilt, I track the degree days in Celsius because that's what the research has been done with. And yeah, I think the U of M extension just tends to go, okay, it's April, boom, we shut off oak wilt, even though like the degree days might be saying you'd still be safe-ish, and it's a gradient. It's not a binary thing necessarily. There's gradations of risk. Like, it's not like with anything in life that there's a binary black and white risk. Like, your tree just went from, safe to like hazardous just because of this, or there's like a dimmer switch on it. So, we have this kind of dance with the U of M information, and I've been in communication with Extension too about, like, they're saying optimum time to treat for preventing EAB is mid-May through early June. Well, based on what? Based on adult emergence, growing degree days? Yeah, but you need a full crown of leaves to pull emamectin benzoate into a tree, and that happens from late May through end of September. So the information posted on there has nothing to do with actual practical application of when it's done. But then that creates mischief for clients who read that and say, you're doing it wrong because the U of M says this, and you're coming out doing it like in the middle of July. So yeah, there's some work to happen to align what we're up to there. So yeah, thanks Lauren for putting that in there. Oh, great luck with horticultural soaps. Spinoza said, yeah, timing is everything. That's the thing. Yeah, scouting. Hopefully you've got enough people to scout everything and thoroughly enough to stay on top of that. Because yeah, the biological biorationals, everything is trade-offs, you know, like you can reduce the footprint of chemicals, but it usually requires more timing and ability to respond more quickly. Okay, there are beneficial insects other than pollinators, yeah, differentiated from insect pests. Yeah, there's a whole other talk that we could go into. You know, I am with you on that. Like pollinators are not the only things we're trying to protect, like lacewings, lady beetles, all those kind of things. And that's actually part of in the health triangle, like in the environment corner of that, put more diverse plantings in the landscape so that you have alternate food sources, nectar sources for some of the beneficial predators as well. So yeah, I kind of push back on pollinators being the sole focus of everything. Oh, 1157. Sorry if I'm just blazing through these. Oh yeah, power washing Magnolia scale. I've actually seen people take a toothbrush and scrub Magnolia scale off their trees effectively. If you have the time and inclination to do that, it works. But on a full company scale, not like our techs are going to go out with toothbrushes, but the individual homeowner can do that. Say again, whose research points to those impacts of biochar? Glenn Percival in the UK. Really, I think some of the best work in arboriculture right now is coming out of the UK. I would love to just take a month or 6 weeks and go around England and have a list of people to hang out with and dive into their research. It would be great. Yeah, Dr. Glenn Percival. Thanks, Ryan. Oh yeah, U of M Extension did biochar trials, did not find benefits to continue. Yeah, the thing is with biochar, you can only add 5% by volume per application. You know, if you put in too much biochar all at once, it's so absorptive, it locks up nutrients and the trees go chlorotic. But we're using it in all of our soil amendments that we do with the air spade and mixing in organic matter. It's just part of our standard formula. Exactly, I've run into that as well. Oh, I'm assuming you're talking about the extension information not matching up to when we're actually doing treatments, etc. Yeah, thanks, Lauren. And what's that link there, Ryan?
Yeah, Kent. So we're kind of coming up on time here. I just posted that link. So if anybody needs ISA CEUs for this session, they qualify for one CEU. You can just fill out that form at that link. This session was recorded and it will be posted sometime, hopefully by Monday. And you can find the link to the recording in the weekly MnSTAC email. And it will be posted at our U4 YouTube page. So, but with that, I think in respect of Kent's time, that we will end this session. Kent, thanks so much for doing this. And like I said, bonus points for presenting while on vacation. I think this was it's a great talk.
Thanks. Yeah, it was really a blast to come in and get a chance to put this stuff out there. I hope it's helpful for everyone. And you can email me at Rainbow Tree Care, khonl@rainbowtreecare.com. I should have put that in there. Sorry. I can throw it in the chat if anyone needs to reach me.
Great. Well, thanks everybody, and have a great rest of your day.